Let's go ahead and get started. So it's my great pleasure to introduce Dr. Olaf Sporns today. So Dr. Sporns is a Robert H. Schaefer Chair, a distinguished professor and a provost professor in the Department of Psychological and Brain Sciences at Indiana University of Bloomington. He's also co-director of the Indiana University Network Institute and holds adjunct appointments at the School of Informatics and Computing and the School of Medicine. He received his PhD in neuroscience from Rockefeller University, and he joined the faculty at Indiana University in 2000. So Dr. Sporns has loads and loads of honors and awards, and some highlights include the prestigious John Simon Guggenheim Memorial Fellowship. He's a fellow of the American Association for the Advancement of Science, and he has received the Patrick Suppas Prize in Psychology and Neuroscience Award by the American Philosophical Society. Dr. Sporns has authored over 200 articles and currently has over 51,000 citations. He's also authored 2 very influential books, copies of which can be found in several faculty members' offices, including mine, and several others review as well. Dr. Sporns is very well known for his fundamental contributions to networks and connectomics. He studies diffusion connectivity, functional and effective connectivity, and he uses graph theoretical techniques, and he uses them to study fundamental cognitive neuroscience questions. So with that, it's my pleasure. So please give Dr. Sporns a warm welcome.
Thank you very much for the kind introduction and for the invitation to be here. It's great to visit a neighbor, so to speak. After 18 years at IU, it's my first trip to OSU, so very happy to be here. Had great talks this morning. Thank you for your time. So as Zeynep mentioned, my work of the past 10 or 15 years largely has unfolded in the intersection of neuroscience and network science. And I want to give you an overview in my talk today about what some of the current approaches and ideas are in the field, but also especially in my own work in this area. When I say networks, what I really mean is I don't use the word informally as it's often used in neuroscience as a circuit or whatever, but really as a formal mathematical model, a way in which we can break down a complex system such as the brain into elements and interactions, and thereby create a map of what we call its topology, the relationship of elements to each other as part of a global network architecture. Networks are very commonly studied, have had a tremendous history the last 20 years in terms of new approaches, new tools becoming available, in part driven by more and more data coming in the social sciences and technology, the internet, and increasingly in biological systems. A couple of examples you see on the left is a blog, a reblog network. You can imagine a political blog network. You can imagine what the colors red and blue refer to. And on the right, a systems biology graph, where each of the dots is a protein and the interactions are represented by the edges between them. And I want to make the case today that these more formal, more mathematical, more computational network approaches can also make inroads into understanding the structure and function of the nervous system. So first, a few words about introduction to the networks, terminology, motivation, etc., before I go on to the more subject matter related talks. So one thing that is motivating the growth of network science approaches in our discipline, neuroscience, I believe, is the increasing availability of data. And it's data that is increasingly rich, and it's increasingly about interactions. It's data, it's relational data, as we call it. It's data about interactions among molecules, among cells, among brain systems and areas, and even in the social setting among individuals. All of these data sets that are becoming, many of them that are becoming available to us are very naturally falling under the umbrella of network science, can be analyzed, can be modeled, can be understood using the tools and methods of network science approaches. In the middle of this graph, which is deliberately a riff on a very famous illustration coming from Terry Sinofsky's work, you see the large space occupied by Connectomics, which is a systematic, comprehensive mapping of relations among neural elements from individual neurons all the way up to brain regions either structurally or functionally, with a variety of tools, from neurophysiology all the way to functional neuroimaging. What I want to focus on in my talk, obviously, is fMRI and diffusion imaging, but first I want to give you the broad framework here of what we now call, what is increasingly called now network neuroscience, again driven by the availability of data from genomics, proteomics, and interactions among molecules all the way to social interactions. All of these different data streams flow into a common framework of networks and topology and graph theory that you can see at the bottom, a few iconic images here of networks and how we might analyze them. I'll unpack this for you in just a second. Network science gives us very many descriptive ways of classifying and recording the structure of graphs. Here are a few of them that I'll mention in my talk now and then. These are the very simple ones that we use in neuroscience abundantly. I should mention graph theory is an extremely elaborate field of mathematics. Textbooks on graph theory easily run 500 or 600 or 700 pages. I typically get to the first 10 and then I'm done. And graph theory is very widely applied in technology, in information technology, in social systems, Facebook, Twitter, you name it. It's a very sound mathematical framework. We do not, as neuroscientists, have to be afraid of it. It is actually a very well-developed discipline with 100 or more years of experience behind it. We use in our work most often are very simple descriptive measures, such as the node degree. That's the simplest thing you can record. It's actually just a matter of counting how many connections are attached to a given element in your network. It turns out it's an extremely powerful predictor of how important a given element is in the overall functioning of the system. Very simple though. We can go to clustering coefficient, which simply quantifies if you have neighbors in a network, things or elements you're connected to, how well are they connected amongst each other. That is a measure of cliquishness or clustering, decomposing of networks into motifs, and then the very important concept of path and path length. In brain networks in particular, the concept of communication, I think, is central to understanding how they work. Communication meaning exchange of signals, flow of information among neurons and among brain regions. Now, the brain is not fully connected. Not every neuron connects to every other neuron, obviously, and not every brain region even connects to every other brain region. So many communication events have to unfold along paths that involve multiple intermediate steps. The length of these paths, called path length, is a important measure that we keep an eye on and we will return to in a few minutes later in the talk as a way of assessing or quantifying how close two brain regions are to each other in terms of their communication distance. At the bottom of this, there's two approaches to decomposing graphs into smaller units that can be described more efficiently. On the left side is modularity. One of the most common ways of decomposing a larger network into smaller components is to find clusters of densely connected elements that are weakly connected amongst each other. That's a way of clustering or segmenting components, communities out of a larger network. And critically, when we do that, we define these partitioning schemes that carve up a larger graph into a smaller set of components. We also identify simultaneously those units, those elements, that are connected in such a way that they connect communities to each other. This is the so-called hub node right here. You might imagine if that particular element in the network is attacked by disease process, by injury, by anything happening to it, it has a disproportionate effect on the communication capacity of that network, effectively disconnecting it. This is why hubs are of clinical importance, again, a point I come back to later in my talk. On the right, at the bottom, is another organizational feature that we've been fond of in the lab, and that is the so-called rich club. Once we identify highly connected hub regions or hub elements, we can also ask a simple question, which is how many connections do they make amongst each other? Are they talking to each other more so than expected by chance, where chance is a null model we compare to? If that's the case, we say, or it's been said in the field, it comes out of social sciences, that the network has a so-called rich club organization. Rich meaning not money, although in social systems quite often that flows into it. Rich here means many connections, and club means connected amongst each other. In social system, social systems are a tool to maximize influence. In the brain, we think perhaps a tool for integrating information. So modules and hubs and rich globs are topics I will come back to repeatedly. A very important part of our work is to extract brain network data from observations. That's a non-trivial, very much a non-trivial exercise. In principle, we can extract network data from almost any set of observations. In fact, we have worked with people in invertebrates, vertebrate recordings, as well as in non-human primates in the rodent brain. I'll focus here on the human brain in this diagram because that's, I think, of most interest to everybody here. There's two different workflows here, schematically illustrated. On the left, you see the construction of a structural network, a network of relations among elements, brain regions in terms of the anatomy. The first step here typically is a parcellation of the cortical and subcortical gray matter into nodes. These nodes become dimensionless points, and we assess the probability or the density of connections among them, sometimes also their myelination status. That eventually gives us a lookup table, a square matrix. Think of it as a big Excel spreadsheet. Within rows and columns are our elements or our brain regions, and the entries in the matrix refer to the existence or the strength or the direction of a connection between them. Diffusion imaging and cartography is a tool of choice in non-invasive human imaging. It has its issues, to which I'll turn later. And it is this representation that we proposed in 2005 should be called the connectome. It may surprise you that the connectome at the time wasn't even a word. I'll tell you the story that when we first wrote the paper, I looked up connectome, Googled it, wanted to make sure we're not stealing from somebody else. Didn't get many hits, actually 9 hits altogether, all of them unrelated to the brain. One was a dating website, Connectome.org, which has since gone out of business, unfortunately, but the connectome has not gone out of business, and it's grown tremendously. And in fact, when you Google the term now, you'll get hundreds of thousands of hits. So it was a concept that, for reasons still unknown to me, had some impact. On the right is a different workflow that ends up in a different representation of brain networks, and that's functional connectivity or functional networks. Operates a little bit differently. Typically starts with recording sites or voxels if we are in the fMRI world, and then wiggly lines, which is time courses, bold signals going up and down and sideways. And then from that, using time series methods, often as simple as cross correlation in the case of resting state fMRI, we construct a correlation matrix or some other matrix of statistical dependence among time courses. That is by its nature typically a full matrix, so it's not sparse as the anatomy is naturally, but it's a full matrix. Every entry in that matrix has a non-zero value to it and can be positive and negative, and can be thought of as a network representation. And the nice thing about this dual workflow is we can do this with MRI data in the same subject using both of these pipelines, and then come to a combined structure function analysis. Converge onto a representation of the underlying wiring, if you wish, and on top of that, a representation of the statistical dependencies, presumably a result of the wiring, that report on coherent activation and co-activation across time. One thing I always like to point out is that structural and functional connectivity are two very different animals, two very different types of ways of looking at the brain. Yes, there's commonality, both in our usage in our lab in the field now. Both are networks, yes. Both have topology, that is, we have edges and connections and their interrelationships. Both have multi-scale topology, there is structure at different levels of scale, and both have community organization, typically clusteriness or cluster architecture. But there's also many differences, and these are important to keep in mind when applying network science tools to such data. Structural connectivity is physical and material in nature. It is verifiably there or not there. You can poke it with your finger, so to speak. It is actually physical and material in nature. There's a finite number of connections. It can be a very large number, perhaps 100 trillion in the case of individual synapses of the human brain, but it isn't ultimately a finite set. Yes, there are complex attributes such as strength, conduction speed, etc. And there are changes across time, typically on a fairly slow time scale, we think, development, growth, maturation, plasticity, etc. And it is this underlying structural skeleton that I think of as shaping the flow of neuronal signaling events and communication in the brain. Okay, that's how I think about it. Functional connectivity is very different. It is statistical and dynamic in nature. It is the here and now of how the brain is responding and the relationships among its elements, statistical dependencies, which we can evaluate with a very large number of approaches, from cross-correlation to coherence, to transfer entropy, to Granger causality, to whatever you want. Okay? There's a large and perhaps inexhaustible set of configurations that a given brain can go into. Some will recur over time, but others will not. Complex attributes, again, rapid changes on the order of hundreds of milliseconds, perhaps faster even. With MRI, of course, we can't get to that time scale, but certainly faster changes, rapid changes than we see in the structural anatomy. And I think of these dependencies that we observe with resting state fMRI, for instance, as the outcome of all the signaling events that have occurred. It is sort of an end product of the brain having its conversation that you can't have access to directly because you don't see it. It's the bull response after all, which is that, unfortunately, it's not as direct as we want it to be. And the ups and downs of the bull response are the outcome of those signaling events that are orchestrated by the anatomy. Okay, so we have to distinguish these two. Importantly, even though both these types of connectivity are often summarized now as connectomics, or fall under the blanket of people call connectomics, graph theory tools, for instance, are much more straightforward to use for structural connectivity, for wiring, for sparse wiring connectivity patterns. They are much less straightforward to use and more difficult to interpret in the case of functional connectivity. Functional connectivity, the way it's used in fMRI, is a correlation matrix. The correlation matrix, all the edges, or many of the edges, are interdependent. They're not independent of each other. That means you cannot construct null models the way you could do in a wiring diagram by rewiring edges, for example, as many people do. So there's a caveat here. Graph theory, I'm certainly all in favor of it. I love it. It's a great framework to use, but we should not indiscriminately use everything that's in the toolbox, our toolbox, and throw it at all the data that we can get our hands on, because we will get results back. The algorithms are innocent. You give them something and they will give you an answer, but it may not be as interpretable as you might think it is, especially with functional connectivity. I think we have to be particularly careful there. One thing that works for both domains equally well, I think, is detection of communities, modularity, detection of critical nodes as a result of the partitioning scheme that you adopt. And in fact, community detection has been massively successful in functional connectivity. Resting state networks are nothing else but network communities in functional connectivity matrices. Okay. Right. Before I go to humans and the relationship of structure to function in the human brain, I want to take a quick survey of animal models and studies that we've done and other people have done over the years to look at connectivity patterns in model organisms. Why? Because these are all studies done without MRI in the mix. And many of my neuroscience audiences, this may not be true here, are somewhat wary of imaging. You notice this? Sometimes there's a bit of a hesitation there. Well, imaging. So the next 10 minutes, no imaging. It's all done with tools that are certifiably ground truthy, EM reconstruction, track tracing, what have you. Okay, that is does not inherit the cynicism that fMRI sometimes triggers in people. Okay, so we have a small number of species for which over the years people have constructed connectivity or wiring diagrams from invertebrates like C. elegans and Drosophila all the way to non-human primates and humans. And in fact, as an aside, we are in the process of working with a group overseas to look at multiple diffusion imaging data sets from various mammalian species, over 100 of them, to look for patterns in the way the wiring diagram may have evolved. This sets up the possibility to do comparative connectomics across species. C. elegans, the nematode with around 300 neurons. This work was pioneered by Sidney Brenner, who passed away just about a week ago, Nobel laureate, and someone who has invested a lot in understanding C. elegans development in neurobiology. Coming out of their work in the 80s, conducted largely manually reconstructing EM micrograph sections, was this wiring diagram of about 300 neurons with about 3,000 chemical synapses between them, shown in red, and then some gap junctions shown in gray. That this network, even though we've had it now in the field for 20 or 30 years, still is being looked at from all sorts of different angles in terms of its architecture, its functional properties, its predictive properties, its relation to behavior, etc. A few highlights here from previous studies that have been published over the last decade or so. There's modular organization. It's high clustering and short path lengths, so neurons tend to have a cliquish neighborhood organization. If neuron A and B are connected, if neuron A is connected to B, neuron A is connected to C, B and C are very highly likely to have a connection as well. There is a broad degree distribution. Not all neurons have the same number of connections. Some have somewhat disproportionately more. There are modules interconnected by hub regions, and the highly connected neurons, the rich neurons with many connections, also form a club in terms of having connections among them, despite the fact that they are widely spread out in the worm itself, in the head and tail region. Also, they maintain dense connections amongst each other. This data set is fairly unimpeachable in the sense that it's EM reconstruction. I believe it's quite possible that it will be revised in the future, as this has never been done again since for 30 years, and we have now new technology, I'm sure, so we revisit it, but we can be fairly certain we are not getting a whole lot of false positives or false negatives in this particular data set. In Drosophila, I had the privilege of working with some colleagues in Taiwan and at UCSD over some years to look at the Drosophila connectome at the level of local processing units. So these are like brain regions in the Drosophila brain. Again, we find evidence for modularity, evidence for rich club organization, and we can identify through this partition that you see here some of the highly connected and critical brain LPUs, or brain regions, that are important for organizing, as we lay out in the paper, for organizing integrative behavior. Moving very quickly to the mouse, thanks to the Allen Institute, we have a first draft of the mouse connectome. We had it for 5 years now. This is constructed from fluorescence imaging of track tracers injected into the mouse brain and automatically mapped. And subsequent analysis, network analysis by Mika Rubinov and co-workers, has shown the existence of highly connected modules consisting of brain regions that we believe are functionally related to each other, such as visual and auditory regions that you see here in blue and orange respectively. It's just the cortex you see there. And then highly connected, these red streaks in the matrix are highly connected, rich brain areas that interconnect these modules to each other that have connotations such as the anterior cingulate, the orbitofrontal cortex, parts of the thalamus, and the basal ganglia. In my own work with Larry Swanson at USC over the last few years, we have gone through different subdivisions of the rat brain, starting with the cerebral cortex. The way this work proceeds is that Larry and his co-workers are methodically combing through the published anatomical literature on rat brain, with a variety of which consists of studies that have been carried out with a variety of tracer molecules and histology. So it's invasive and thought to be quite accurate. And he then makes these essentially these big data collections where each element in this matrix again refers to a connection between two regions of the rat brain, and the strength of the connection is assessed by combing through the literature, the existence and the strength. So it's a collection of, in this case, I've forgotten the exact number, but I believe it is over 1,000 individual papers that have gone into this matrix. with many of the connections reported here, some of them having multiple reports behind them. So it's a different way of collecting the data through literature survey and collation. We once again find these organizational features, modularity, hubs, highly connected regions that are also interconnected amongst each other into a rich club. We look at the location of these modules on the cortical surface. This is the so-called flat map representation that Larry is very fond of and has pioneered in his work. We discover immediately that these modules that are based entirely on topology, on density of connections, occupy contiguous real estate, if you wish, on this flat map. So they correspond to regions that are spatially close and contiguous on the cortical surface. This indicates that, in fact, there's a connection here between spatiality or geometry, physical embedding of these regions, their distance from each other, and the topology they actually maintain. This is true also in primate species, where if you have two brain regions next to each other on the cortical surface, sharing a common boundary, ballpark estimate, depends a little bit on where you look exactly, but the ballpark estimate, the likelihood of them being connected physically through a pathway is 90%. This is obviously not true if they are far apart from each other. So there's a strong aspect of geometry. In our later installments, I sometimes jokingly call it a franchise because we're publishing about a paper a year now on different subdivisions of the rat brain. Have the thalamus coming out hopefully later this year. The hypothalamus just appeared last week. This is the bilateral cerebral cortex, so two hemispheres of cortex. And interestingly, what you see here is, in my view, the only full collection yet of callosal connections in a mammalian brain. So this is the, I have a hard time showing it here, but if you see the upper right-hand quadrant and the lower left-hand quadrant of this matrix, you see that diagonal streak running through there? That diagonal streak you see are homotopic connections between brain regions on the two sides of the rat cerebral cortex that are corresponding to each other. Like I said, these are connections, by the way, that are quite difficult to map in humans because the callosum is like a funnel. Your streamlines go into it, and if you have if you go off, you come out the other end with a little bit of an error, you may go anywhere you want to on the other side. It's very hard to get callosal connections mapped accurately. So this is a very nice way of studying callosal connections in a model organism. And what we found is that 32,000 connection reports going into this, a lot of work collating this expertly. The matrix is almost entirely full, 95% data coverage, so there's only very few blank spots where there's no data available. And of the homotopic connections, for instance, of the 77 possible homotopic connections among cerebral cortical regions, only 50 exist, about 2/3. And the heterotopic connections are quite sparse. Another rule we discovered here is that all heterotopic connections, all connections going through the callosum from area A to another area B on the other side, have a counterpart on the same side. There's not a single heterotopic callosal connection that does not have an association counterpart, a counterpart on the exact same hemisphere. The reverse is not true. There are many association connections that are not having heterotopic Commissural connections. So that's an example of how we can discover some, I think, quite interesting rules about how this is playing out in these model organisms. Whether this holds for primate brain, we don't know. This is the second to last installment of the franchise, which is the rat Endbrain, which is the complete connectivity matrix of the cerebral cortex on both sides with callosal connections, and the basal ganglia or cerebral nuclei on both sides with all interconnections added. And when we finally came to write this paper, we had simultaneously worked out a scheme for doing community detection in a multi-scale manner, in a principled way. What you see here, these four main modules are the modules that we believe are most stably detected when we do community detection using network science tools. but they are not the only modules that matter in this architecture. I'm very much in favor of looking at organization of a network like this. It's a multi-scale, inherently multi-scale effort. That is, there are larger communities within them, we have smaller communities within them, we have even smaller communities, like a Russian doll kind of thing. And with this new technique that we've developed with our colleagues in the computer science department, we can come up with a hierarchy that you see on the right, a hierarchical tree, essentially a dendrogram of modules clustered within modules throughout the entire organizational scheme. All of them surviving significance testing, so they are certifiably modules that are real in the statistical sense, at least, and they can now give us a much more fine-grained understanding of which areas of the rat brain belong together and how strongly they affiliate with each other. Okay. Affiliation in a community is a graded property. It's not something which is all or none, in my view. This is the tool that we have now this year also used for the first time in functional neuroimaging and resting state recordings in a clinical setting. And I'm very eager to disseminate this further. So if any of you want to use this, please let me know. We will be happy to send you the scripts. Finally, although in terms of history, this was where my work started, the non-human primate, where we also have had for a long time databases that record anatomical projections among macaque cortical regions. We and others have analyzed these over the years. As the database has grown, the connectivity matrix has expanded as well. And just one highlight here from a few years ago now, working with my undergraduate student, Logan Harriger. He found that macaque cortex also shows evidence of witch club organization, highly connected, diversely connected brain regions. You see some of the designations here, 9 and 46, I believe that's prefrontal cortex, LIP, that's parietal, 13A, 32, I think that's cingulate, 24, TFDH, that's temporal. They are widely dispersed across the macaque cortical surface. Here are their locations, and they're also densely interconnected amongst each other, more so than expected by chance. This is a feature of topology of brain organization that I personally find quite non-trivial. Why? Because to achieve this rich club organization, the brain, in a sense, had to invest a lot of space and material and resources to create these long-distance pathways that link these areas to each other. Just as an aside, we can do, I'll skip this. This is the cellular stuff, which shows, by the way, rich club organization as well. Okay, let's go through it. So my brief run through these model organisms was to show you that with a variety of different measurement techniques, from EM to track tracing to literature surveys, we can construct connectivity diagrams, connectomes for species and for parts of the major subdivisions of the nervous system that have some set of overlapping properties. Broad degree distribution, that means there is most areas, most neurons have a moderate amount of connections, but there are some which have much more diverse, much more widespread connections than the rest. There's specificity to connection profiles. In every system we've looked at, whether it's neurons in C. elegans, whether it's brain regions in any subdivision of the rat cortex, every region has its own unique set of inputs and outputs, which we believe in a network setting is critical for maybe not determining, but at least influencing that area's functional specialization. In a network setting, who you connect to, either by acquiring inputs from or delivering outputs to, that defines your functional contribution. I think, and I may be quite wrong about this, but I'll say it anyway, I think that a lot of the computational capacity of the brain is not so much in the microcircuits and what they do, although they do remarkable things themselves, but the capacity of a brain region to contribute to a function is not so much encapsulated in the microcircuits in the area itself, but rather it is inherited from its relationships to other regions in the brain. A very different view of How you think about the brain, and classical view is you have these modules which internally perform computations, and the output of the computations are sent across to other regions. I think we need to flip that scheme upside down and say it's really the network as a whole that makes the computation happen. I digress. Modules and interconnected hubs that form a core or a rich club are also a common feature in all these different organisms. This in itself should strike you as either remarkably boring or remarkably interesting. Think of it, we're finding, I had to give a talk some months ago in a meeting that was about what makes humans special. I don't know why they invited me, because I really had nothing to say. When it came to the topology of brain networks, we have yet to find a particular architectural feature that's unique to humans. There is none. In fact, C. elegans has all architectural features, the big ones, rich club, modules, hubs, dense, degree distribution, heavy tail, that also humans have. Okay. And so that sets up a question, why is that the case? And it turns out, in my opinion, one of the big driving factors here is that anatomy, because of its physical nature, it's real, it has to actually be built in space, consumes resources like volume, material, energy that are not unlimited. That sets up a competition for these resources among connections. Most connections, therefore, must be short in length. That wiring minimization constraint has been recognized for a long time in the field. It goes back all the way to Cahal, who has written about it. But it cannot be the whole story when you think about it. If all the brain wanted to be was maximally cheap, there'd be no connections at all. That would not be a good brain. That would not be a brain that could actually support behavior in a way that actually makes sense. So we need to have some features of the organization that are contravening the drive towards wiring, and those are features that we have our eye on because we believe they are particularly important for global functioning of the architecture. They include things like hub regions, which tend to be costly because their connections are widespread and long, and rich club organization for the same reason. Together with Ed Gilmore, my longtime colleague, some years ago, we wrote about this and came up with this economic idea informed by trade-offs, thinking about these different forces of wire minimization and efficiency, as we call it, which is good functioning, as opposing trends that actually operate against each other, and we might be able to understand something about how the brain is organized based on that. If our brain was maximally cheap, all connections would be short. We get the brain that's on the left. That's a cheap brain in terms of wiring cost, but think about moving information from the back to the front. Well, that would be a long journey. And on every step there'd be noise, corruption, there'd be delays. It's not good. On the right, random topology, ironically, is the best way of slushing information from one end to the other, but it is extremely costly to wire up. It is astronomical, actually. The brain does not fit in your head anymore if you wire it randomly, and that would not be a good thing. So we think the topology we actually find is a mixture of these two. There's largely wiring minimization at play, which gives rise to communities, functionally related brain regions, taking up space that's in close proximity next to each other, perhaps on the surface. And on top of that, an admixture of long-range pathways that are organized in such a way that they keep the whole brain coherent and integrated. That's the rich club organization. And that's the efficiency that the rich club performs. We have been exploring that idea in various directions, using computational simulation, using morphospace, embedding of networks, etc. But that's a different topic. Turning to the human brain now, when I first got into into neuroscience, our knowledge of human brain anatomy was quite limited, and limited really to postmortem dissection and the kinds of illustrations you see here coming from a famous histological atlas of the human brain. It's actually a photograph of a postmortem brain that's been dissected very carefully to expose the interior of the white matter, and you see some of the fascicles and tracts running back and forth, connecting long-distance brain regions. There's a distinct there's an advantage to this because this is in some ways the ground truth. You take a photograph of it, people can't accuse you of, having fudged your data or whatever. But there's a big downside, which is this person is dead, and you can't do this on a large population. And you can't bring them back to the lab and look at their behavior or cognition, and it's highly invasive, obviously. So this is not good for the purposes of really studying individual differences and relations to clinical disorders, etc. And for that, we now have a technique that most of you will know, diffusion imaging and tractography, which gives rise to these colorful hairballs or spaghetti diagrams that often illustrate the covers of textbooks and such, that are not direct visualizations of tracts, but are actually a model. This is actually a model that you see. This is an inference based on a complex diffusion signal coming from the white matter for the most part, and a model put on top to explain the three-dimensional distribution of the diffusion that you see. And like all models, there's assumptions going into it. There are free parameters. There are issues of resolution, reproducibility, etc. So there's many issues with this technology. However, I am very much a glass half full person. This is the only shot, the best shot we have right now of non-invasively imaging some large-scale aspects of human brain anatomy. And for that reason, if none other, the technology deserves to be pursued, and is being pursued in many places. It's getting better and better, and many of the things we see have analogs in so-called ground truth techniques such as track tracing, as we know from studies in model organisms. So this is the technique that I became familiar with in the mid-2000s through the work of Patrick Hagman, who became my very important collaborator at the time. And together with Patrick, we conducted the first, if not, probably one of the first, I'll say, to hedge my bets, studies of connectivity studied with diffusion imaging and tractography in human subjects. As an aside, this paper was carried out on 5 individuals. You cannot publish a paper or imaging of any kind with 5 subjects, unless you have a very good reason for why there are only 5 subjects. But so we really started small with this. The things that we found in our initial analysis that you see listed here are perhaps now looking familiar to you, because they have subsequently also been found in all the animal systems, animal models that I showed you earlier. Okay, so the whole story chronologically took its origin here, and then people went to C. elegans, Drosophila, rat, mouse, you name it, to look for these features in those data. I've shown you these earlier. And happily, those features are quite robust. These types of studies have been carried out in humans now with much larger cohorts, with much more modern technology over and over again since we published this first report, and have all been replicated many times. Just a few words about the Rich Club, because that was one feature we didn't have nailed down right at the beginning. Came to us just a few years later. Just to unpack one more why we care about this, why we should care about this a little bit as we talk about brain organization. So we have on the left some modules, slightly illustrated, densely connected internally, weakly between, and some hub nodes in black that connect the modules to each other. If we lose hub nodes, that's bad for communication. But if we have more connections between the hub nodes, shown in the middle, then we have in some sense, a community or a dense cluster that sits on top of the other subordinate clusters and connects them to each other. And we felt at the time and still feel that this is an important architectural feature apparently shared by all nervous systems people have looked at, from the worm all the way up to humans. Such an architecture creates efficient, short paths between all communities, and it also is a good, perhaps a good substrate for integrating information together. because information can flow into these highly connected hubs and then can be shared in a kind of cortical workspace or dynamic core for informing behavior. Working with Martin Heuvel, we carried out the first Rich Club study back in 2011 on a modest number of subjects, found the Rich Club effect, which is that red curve at the top, which illustrates the excess of connections among highly connected or rich nodes over the null model that we chose. We can argue theoretically based on modeling that if we want to move information around in the human brain from any place to any other place, most of those communication paths have to access the Rich Club. Even though the Rich Club involves only about 10 or 12% of the total infrastructure, if you wish, of brain connectivity, 60 to 80% of the traffic, message traffic along shortest paths must pass through it. At least that's what our modeling suggests. If Rich Club nodes or edges are damaged, another prediction is that this will impair communication and therefore also impair functioning of the brain disproportionately. The latter point is has triggered a large number of clinical studies now with using both functional and structural connectivity that have looked at points of vulnerability and points of damage in the progression of various brain disorders. Focusing here on the left side of the plot, this is a plot of, or an illustration from a meta-analysis by Nick Crossley, in which he looked at around 30 or 35 different disorders, looked at T1 scans from over 22 or 23,000 individuals, and tracked where in those anatomical scans do we see signs of atrophy or of abnormal gray matter organization, and how much in each disorder does that overlap with the hubness of the region that we're observing the damage in. And there's a statistically highly significant bias, not for all disorders, but for many of them, towards damage observed in anatomical scans that impairs apparently more highly connected parts of the connectome. And you can see the range of disorders, the read this in the back, is extremely broad, from schizophrenia and autism to various degenerative conditions, Asperger's syndrome, Huntington's, etc. So there's a lot of emphasis now in clinical neuroscience to leverage our insights into connectome organization to see if we can make sense of clinical observations of physiology and regions that are impaired in disorders with respect to impact on patients in terms of their behavioral cognitive dysfunction. We have written, Martin Heuvel and I have just written a new review article on the subject of clinical connectomics, which will come out in the next month or so, and we've tried to come up with a few sort of broad ideas for how we can conceptualize these different disorders under one umbrella of connectomic dysfunction. Okay, functional connectivity. I'm 15 minutes into this, and only now I'm coming to functional connectivity. So see, my thinking about the brain is very much structurally based. Okay, I feel very strongly, maybe because I was trained as a biochemist, that structure actually matters a lot for function. And so I have the connectome, the ambition of the connectome was specifically directed at anatomical connectivity. But it does raise the question, if we have the connectome shown here on the left in one of those diagrams, how do we get from that, which is relatively static? You know, some people think of it as boring because it doesn't change much over time. It's not boring. To something which is much more flexible, dynamic, and engaged in activity on the right. How does that transformation take place? What you see on the right, by the way, is a movie of someone in an MRI scanner doing nothing in particular, so-called resting state. This is an early rendering of this by Justin Vincent, 2007, when resting state was a dirty word, or two dirty words, that a lot of people did not believe had any value at all. And now look at what happened. And what you see here is ups and downs of the bold response, projected onto the cortical surface. Each of the orange spots, yellow spots are bold activation. Incidentally, the amplitude of these activations is on the order of magnitude of what you see during task activations, about 0.5% or 1% up and down. So it is not a teeny amount of noise in the background on top of which you have these huge task signals. It is much more like if you have a task signal, you couldn't even see it in this movie. It will be submerged. Okay, so this is what's going on in a person's brain. Awaken alert, slightly sped up for illustrative purposes. So how do we get from the left to the right? What do these two things have to do with each other? Now, the way we study functional connectivity, many of you know this. We take frames from movies like this. We make cross-correlation matrices. We cluster them, and then we proudly give these clusters that we deserve names, such as default mode, somato motor, visual, etc. And as we all know, we are primates after all. When we name something, we really know what it does, right? We can now throw these names around and everybody's happy. If you have a study that finds something in a default mode network, yay, everything is good. This resting state is remarkably reproducible. If you take enough data, 10, 15, 20 minutes, and you do this in individuals in large cohorts, you get very reproducible maps of resting state networks, clustered, coherent communities of interacting brain regions. 2 different studies back-to-back carried out by Randy Buckner on the left, I believe, and Power on the right. Probably Randy showed the same picture a couple of days ago here. These are very highly reproducible across subjects and also within subject across different scans. They're also sensitively modulated by individual differences, and this is where it gets interesting because we can assess individual differences using this organizational scheme as our marker. That's where we come in as computational neuroscientists. Where does that come from, right? And preceding resting state networks, really, we worked in computational neuroscience, my grad student Chris Honey and I at the time, on implementing models of functional connectivity in computational simulations. These simulations had 2 main ingredients. On the one side, a coupling structure, which is the anatomy you see here. This is the connectome of the macaque visual and somatomotor areas coming from track tracing studies, a binary matrix. And on top of that, we put biophysical equations that tell us what brain regions or neural populations in brain regions do across time, the ups and downs of the voltage signal in excitatory and inhibitory populations of neurons. These equations come from classical neurophysiology, Nernst equation, conductances, etc. Many parameters here, but we never vary them because they're given to us from empirical studies. The only variable that we ever changed now in our investigations is the overall coupling strength of the connections. We then can pass the time series that we can simulate. It's essentially now a physics simulation, coupled differential equations, ODE solver, like in MATLAB, you can invoke and you can generate time series. You can pass these to another model, which is a hemodynamic neurovascular response. You can make synthetic fMRI responses out of this, which we've done. Much cheaper than running a scanner. And you can then get functional connectivity matrices of bold cross correlations that you see here. At the time, we had no way of comparing to empirical monkey data. We did not have any. But years later, Yasushi Miyashita came to us from the University of Tokyo. We had some data he wanted to compare, we did, and the relationship is not perfect, but it is also not 0. So we have perhaps in our hands, this line of work has since been pushed forward by many other labs in many different ways, with many modifications and improvements. But basically what you're looking at is a generative model of where functional connectivity comes from. It is a coupled it is the result of coupled interactions among neuronal populations running through the connectivity matrix, which is the connectome. The great thing about this computational approach is you can perturb the matrix, you can make lesions, you can simulate the effects of stroke or brain disorder, and then you can try to predict functional connectivity patterns resulting from that. That's just one application. because I'm so far along, I'll skip this. This is another wrinkle on the same story pursued by my postdoc at the time, Joaquin Goni, and yet more wrinkles by my postdoc Vladislav Micic and Vic Betzel, my PhD student. We went in a direction of implementing simpler and simpler models to predict functional connectivity. Other labs have gone the opposite direction and tried to make the models more code realistic with integrate and fire neurons, with local circuits, layer structure, etc. We went the other direction. The bottom line is that we, even with very simple, almost analytic models of spreading that come to us from social sciences, we can actually make useful observations and predictions about the organization of communication events and resting state networks in the human brain. So the way I now think about this is encapsulated in a recent review article spearheaded by my former student and postdoc Andrea Königsberger. The way we think about the whole framework here is like this. I start typically with a consideration of the anatomy, because it is for me the bedrock of brain function. It is, this goes back to Cajal, who gave us that, incredible amount of insight back in the 19th century. In some ways, we are building on that in a new direction now. For me, anatomy is fundamental. And on top of that, we then have signaling events. Neurons, for the most part, that we care about really can only communicate through external pathways and synaptic connections. And there's some other indirect ways. Yes, there's neuromodulators. Yes, there's hormones. There's slow signals. But for the purposes of fast communication, it's synaptic connections. That sets up what we call communication dynamics, which is the ups and downs, the flows, the spreading patterns of these signals on the anatomy. And the end result of that are time courses. Remember one thing, when you measure functional connectivity, you are not measuring the connections themselves, you are measuring time courses. You are seeing ups and downs of the Bowl signal. From that, you try to infer, this cross-correlation, which is a poor inference tool, but okay, you try to infer what the connections are. We actually do not see connections directly. We cannot. We don't see signaling events. We don't see causal relationships very clearly, certainly not with fMRI. So we're getting functional connectivity, which is really an outcome measure, if you wish, of what's happening at the signaling level among these interconnected neuronal populations. That's how we think about it in the lab, and that's computationally the kind of framework that we try to push. The middle thing here, this is a thing that I have not mentioned yet, effective connectivity, in part because it's a little elusive. This is the level that we in the lab feel we really should get to, which is the ebb and flow and the direction and the changing dynamics of the communication events themselves. But these cannot be observed right now with any existing neuroscience technology. This is a black box for us. We cannot see it. Finally, I'll say 5 more minutes about dynamic functional connectivity, if I may, because it's such a hot topic these days. When I worked with Chris Honey on the model of the Macaque cortex, the computational model, it occurred to us that we shouldn't just look at the overall sum total of all the functional connectivity that we observed over a long period of time, but we could also use windowing to look at shorter epochs. When we did that, we found that the status of brain regions in the macaque network as hubs, as parts of communities, would change dynamically and quite fast. The time base here is in seconds, so this is 900 seconds long. What is that? 15 minutes? I can't calculate right now, I'm sorry. Anyway, so these are sort of fairly slow fluctuations that we see across time. Now, mind you, this is a simulation. So one thing that's not happening here, I guarantee you, is respiration, heartbeat, physiological artifact, scanner noise. This is not undersampled because it's actually done on the fast neuronal time traces, 30,000 time points per window, so this is not undersampled. It's a phenomenon that is very familiar to physics people that I've talked to. That is multiple time scales that suddenly materialize when you have a complex dynamic system you simulate. You get relevant events happening in multiple time scales. That was actually the title of the paper, which is often forgotten, even though it's been highly cited. So that was our first observation in some ways of dynamic functional connectivity. I remember taking these plots over to WashU at some point many years ago, and I was told very firmly that no, bold signals are stationary. They don't fluctuate over time. So both functional connectivity is stationary. So as you know, there has been a lot of activity recently in dynamic functional connectivity nonetheless, and there's, I think, mounting evidence that despite the fact that the bold signal is temporarily challenged, let's say, there are still signatures of underlying fluctuations that perhaps are real and perhaps neurobiologically meaningful. Over the last 3 years, I've worked with my postdoc Makoto Fukushima on this, and the approach that we've taken is the following. We're looking at using windowing of dynamic functional connectivity. We look at modularity fluctuations. We're looking at fluctuations in the way communities coalesce and disperse. The overall degree to which a network is modular, a functional network is modular, varies across time, as we find. What Makoto then did is he defined a cutoff above and below which there are significant events that are not explainable by chance alone, that involve excursions, as Rick Wetzel called them, away from the norm, unexpected states that the brain is going into of either high or low modularity. And what Rick and then Makoto did was go back to the time series, cut out those frames of where those excursions occurred, and average them. It turns out there's a common signature to those high modularity as well as low modularity events that is reproducible, in this case, across 2 different data sets. The reviewers really made us go from NKI to HCP and backwards with different postulations and who knows what. It was a never-ending, 10 years ago, we published 5 subjects. This paper, I think, had over 700 subjects in it, and it was still not enough. Okay, so that's how things change. Anyway, Pro Makoto was really burning up a lot of computing time on the study. Bottom line here, and I have no pointer to point to, but perhaps you can see it. In the high modularity cases, where some systems are particularly coherent and well-defined, what we see is that the default mode network and task-positive networks are strongly decoupled, internally quite coherent and strongly decoupled from each other. This is a common signature that we see during high modularity events that occur across different subjects and across different data samples. And during low modularity events, that coupling isn't as well developed, and the anatomy of the brain is much more highly correlated with functional connectivity. So we're seeing more of an alignment between functional connections and structural connections in low modularity events, and the brain escapes from that in some ways. High modularity network states take place. They have a recognizable topology to them, or topography to them, in the sense that task positive and task negative systems tend to be decoupled, which is something that we continue to pursue with collaborators as well as in other applications to this. So the picture that I have here for you at the end is that on the left side, anatomy, structure, relatively stable across time, we think, although there is probably a lot happening in learning and plasticity, especially with white matter. Modules interconnected by a rich club, and on the right, much more dynamic ebb and flow of highly modular, highly segregated, and more integrated low modular states that follow each other during resting state. And of course, on top of that, task manipulation. I haven't talked much about it, but there is abundant evidence from the work of other laboratories now that tasks will modulate functional connectivity patterns, and that is a whole other part of the field that I have not said much about today. Okay, I'm coming to the end. The picture I want to leave you with is of a brain that has many different elements and components at different scales. However, what all of these elements and components have in common is that they're organized into networks. Okay, I'm coming back to my overall theme of networks. The network organization of biological systems, I think, is fundamental. It is how complex systems organize and how they maintain their flexibility, their robustness, and their functionality across the spectrum, from molecules all the way to brain systems. And because network interactions, I think, are so important to consider, and they really haven't been, I think, considered enough in neuroscience until now, because I have nothing else to do. I started a journal a few years ago called Neuroscience with MIT Press, open access, and now in its third year of publication, and you can check it out, look into it, open to all submissions in all model organisms, human, clinical, translational studies, anatomical, functional molecules to brain system, but with an overarching network theme to it. My goal is to create a community of people that talks to each other and also brings in people from fields that we need input from, such as data science and computing. And with that, I thank you again for your attention, and I hope that you got something out of this. Thank you.
I guess I will go first. I'm sure there are many questions, but people are still digesting.
Yeah.
So one word that I didn't hear and I expected to hear is gating.
What?
Gating.
Gating.
Which is one region modulating the connection between two other regions. And so is that visible with your methods? Have anything to say about that? Because it is highly likely that the brain uses that.
Yeah. I have, I mean, and when you say my methods, I mean, network science is what it is. It can look at any network data set in any shape, time series, what have you. And it would see gating events, yes, for sure. If a connection is opened up as a result of the activity of another region, let's say, that could be discerned if the data was suitable for it. Now, with fMRI, I don't know, because gating events of the kind that you refer to typically are unfolding on I think, on faster timescales. So I think we're a little bit flying blind here with, I don't know whether it's true or not, but I suspect that with bold signals alone, we are a little bit impoverished, because we don't have access to that millisecond or 100 millisecond time scale at which these things might occur. Having said that, the techniques and the approaches, the tools, the methods of network science can be extended very naturally to the cellular domain, to multi-electrode recordings, for example, where you have other problems to deal with. You have the fine time scale, but you have a massive undersampling problem. You will never, despite what people might tell you in TED Talks and God knows what, we will never be able to record in our lifetime from all neurons simultaneously of the human brain. That's not possible. So we always have this massive undersampling problem. So that's an issue. But in principle, yes. And I actually believe that we talked a little bit about this in our review article 2018 about communication dynamics. We believe that these dynamic routing schemes, that is opening of communication channels as a result of a third area, let's say, bringing two areas into coherence with each other, sort of communication through coherence, is probably a very important scheme to keep an eye on. And you know, that's something that people have proposed. We could look at it with our technique, yes, but not in bold responses, I suspect.
Thank you.
Great talk. So I was wondering about what your thoughts are on the, so you talked a lot about the adult organisms, right, across different species and so on. And so I'm wondering if, what your thoughts are on the development of Bridge Club across the brain and across different organisms, and whether that might give us some insight into the differences that come out of these cells.
It's a great question, and still to be determined, I would say. One thing that we know of that's the case in C. elegans. In C. elegans, the C. elegans, the nematode is a particularly interesting organism because every cell in its body, including every neuron, has a lineage. That's what made it such a great model organism in the first place. So we know when these cells are born, so to speak, when they're made, they actually all have names. The neurons all have names. You can find them presumably in every nematode. They have little designations. So they have identities and they have histories. And it turns out that rich club neurons, the neurons that end up have many connections and are interconnected amongst each other, are among the first that are made. That's one result from the C. elegans literature. We have considerably less information in humans. I have worked with Patrick a little bit in the early days in developmental cohorts of individuals with ages ranging, for example, between 2 and 18 years. And we were struck by the fact that a lot of the anatomical connections, while their myelination may not be at the adult level, the connections themselves were demonstrably present. So the topology, it sort of stands to reason that ought to be the case, given that most synapses are made relatively, in fact, in overabundance after birth, in the first 2 years, if I understand correctly, and then are pruned back with subsequent period of time. So the anatomy might be in place quite early. Functional networks, I know you're looking at those, are not as mature yet at those early stages. Coupling between structure and function, the degree to which functional connectivity is predictable from structural connections is much lower in our hands in younger kids than it is in adults, for instance. So there are definitely developmental processes playing out. We have more looked at lifespan, which does not include the early developmental period typically, but starts at ages 5 or 6 with the NKI sample, for example. And there are patterns that play out across the lifespan that we and others have looked at, being changes in hub regions, changes in communities, and so forth. The great thing about the general approach is that it's totally applicable. If you have good data, you can answer these questions. It's that we haven't had yet, we have not had, I think you have data, but we have not yet had data from very young kids in our hands to look at this. I should also say that rich club organization is an anatomical feature predominantly. It's something that I would look at from the perspective of the anatomy. I know people have done rich club analysis and functional connectivity, but it's one of those things that I feel slightly it's not quite appropriate. Why? Because the nature of correlations that are dependent upon each other through the time series from which they derive, and you can find dependencies among edges in a functional network that have nothing to do with a topological feature, but more to do with the fact that things are actually just coupled. That makes sense.
What do you think besides just being relay centers, what do you think the role of hub nodes and rich clubs are, and do you think that role varies from species?
Second part of the question, I'm not sure. I think I still like the original hypothesis that we started out with, which is that since there is a need, perhaps, general need in organisms to integrate information from multiple senses, from multiple sources, including internal sources, an architecture like a common workspace. In fact, there's a workspace theory of consciousness out there. A common workspace or a infrastructure that facilitates that essentially makes sense to me as an adaptive feature. It makes sense to me for an organism as humble as C. elegans, as well as for humans. It's ultimately the major challenge of collecting all this enormous rich input and then ultimately channeling it into a common output, which is what your body does. Okay? Whether you're a worm or a human being, that task is the same. So I wouldn't want to speculate on whether it's more important here or there. Now I've forgotten your first question. What was that?
I was just, I mean, you talked about it in terms of sort of network flow.
Yeah.
You didn't talk about network flow, but it was sort of implicit that hub nodes facilitate, information flow from one region to another, which kind of makes it sound more like a relay center than anything.
It could be relay if you don't think hubs internally add anything to the information that they pass on. But that's an open question. I think I could well imagine that there might be hubs that add nothing in particular. They just pass things on, collect and pass on, or perhaps hubs that have more different internal circuitry where something is added on, processed, etc. But I wouldn't. I think there are ways to discriminate between the two, but I have not thought quite as far along as that. The game has been so far to even establish that there are these things. Okay, we have to be. It's still early days. Do we really have hubs? This is a counterintuitive idea when you think about it. Most textbooks will tell you that cortical neurons have 10,000 inputs. Roughly speaking, that's presumably coming from a Gaussian distribution. So it stands to reason that brain regions ought to have about the same number of inputs too, maybe. The fact that that's not the case, we think, is in itself a fact that we first have to really nail down, make sure it's real. Diffusion imaging and cartography is not a good tool to nail things like this down in humans because there's no ground truth. We have nothing to compare it to really in humans. That's where the model organisms come in. That's where all the other work with track tracing is ecology, EM reconstruction comes in because that's a much more, arguably, these are approaches that have more immediate power of persuasion, if nothing else. And that's still a work in progress. So I think of them as relays, some level, but the connections between hub regions may well have particular properties that render them special in terms of the information content they can carry, the capacity to integrate. I used the term integration information a couple of times, and I will confess right now that that's actually a pretty broad term to use. What is that exactly? Okay. We have spent over the years many discussions also with my old colleague Jules Ginoni back in the day in the 90s. What does integration really mean in the nervous system? Is that simply a high order dependency, sort of some high-dimensional probability distribution function that neurons map into? That integration, or is it an active process? And I think when I say integration information, I mean it loosely right now. I think we have a long way to go to really understand what that actually means in terms of operationally, in terms of the physiology. So I'm investing quite a bit of time right now to. I was talking to the students at lunch about it, to branch out and work with people who do physiological recordings to see, first of all, if the types of phenomena that we've seen in human neuroimaging, types of architectures that we've seen, whether they carry over into the domain of single neuron or neuronal population interactions. And secondly, what else we can discover along the lines, because we have a much more fine-grained approach now, what else can we discover above and beyond what we already know from human imaging. This is sort of a new frontier for me I'm very excited about, and that will perhaps allow us to answer the question that you posed.
I saw in your biophysical equation you have potential, velocity, perhaps Q is chemical fusion, I don't know. What do you think is the role of myelin in integrating myelination, if you will, integrating the functional and the structural information.
That's a difficult question. I am. We sometimes take into account myelin related measurements in constructing our matrices, so CFA for the distribution coefficient as an index of efficacy. So we're thinking of it as something that either facilitates or impedes signal transfer. I don't know whether myelination status or what the relationship of it is to, let's say, conduction velocity. This is purely ignorance on my part. It probably is a relationship that's been worked out, but I don't know what it is. We have not used conduction velocity in our own simulations as much as other people have, because my feeling is with the bolt response being so slow, those velocities maybe not as important to consider. But so that would be two ways.
Use potential.
Hmm?
So use potential.
Yeah. Okay.